Okay, so we are live. Welcome back, everyone, to part two of Larry Abbott's talk. So as before, please type in your questions in the chat box, and at an opportune time, I'll ask Larry those questions. Thank you. Over to you,
Larry. Okay, let me share. Okay, so yeah, please ask questions. I think I have less material today to get through, so there should be plenty of time for questions and time for a little discussion at the end, hopefully, if I keep moving. So please feel very free to interrupt with questions. I'm happy that way. Okay, so I promised you last time the sort of second piece here. Which is, how does a fly know which way it's traveling? Last time we talked about how the fly establishes this blue vector relative to the world, so it knows which way it's heading in the world. But I mentioned at the beginning last time that, for example, if a fly is flying around in a breeze, a crosswind, like I've shown here with the arrow, it's not going to be moving in the same direction that it's heading. It may have a drift. And of course, if you want to get from one point to another, the relevant vector is not which way you're heading, but which way you're actually moving. So you would want to line up that purple vector with whatever your goal is for arriving at a destination. And at the end of the talk, I'll mention a little bit maybe how that happens. But most of my talk is concerned with how does the fly compute this difference between the direction that it's headed and the direction that it's traveling. And actually more than that, because we talked last time about how everything, if you want to navigate through the world, all these directions have to be referenced to the world around you. And so once again, we're going to have some kind of a landmark like the sun here. And the angle we're concerned with is actually that angle, not the difference between the blue and the purple angle, or the heading blue. I guess they're both blue, but the reference to some landmark of your traveling direction. So we call this angle the traveling direction. That's what the T stands for, and we call it allocentric traveling direction. That's because it's reference to the world. Not an egocentric direction, which would be reference to the self, to the fly's own body. So that's the math problem of the day. It's kind of a classic physics problem. I'll get into that in a second. But how does the fly do this calculation? And really, you might say this is a fairly simple calculation, but I think it really opens up the whole navigational system for us, because once you realize how flies do vector arithmetic, basically, then you have an in to all sorts of navigational tasks, because navigation is really operations on vectors, adding displacements and things like that. And so to me, at any rate, this kind of opened up the whole method of calculation that's being used in these parts of the fly brain that I talked about last time. I mentioned my collaborators on this part, Gabby Mayman and Chung Liu. This was really heroic and brilliant experimentation by them that I will describe. And I hope you appreciate the beauty and the talent and the hard work that they did, even though I'm the one who's talking. And there are a pair of papers that should come out any day now, actually. Our paper and then a sort of parallel paper that did a lot of the same ideas from Rachel Wilson's lab that will appear together. And then last time I mentioned this gigantic paper. This is a 360-page paper on the anatomical structure, the connectome structure of this circuitry. And, quite amazingly, from the layout of the circuit, these authors from Vivek Jayaraman's group were able to deduce quite a bit of how this thing works as well as, ideas about many other parts of the circuit. So if you've got the courage and the time, it's quite a remarkable paper. So anyway, all this appears in those 3 papers. All right, so how do you go about measuring a fly flying in the wind when you've got to image its brain at the same time? So last time I talked about flies walking on balls, on air suspended balls, so that they could move through a virtual environment. Well, this is the same idea, but it's done by gluing the fly's head to a mount, looking down with a microscope at the fly's brain and looking at calcium signals in the fly's brain that correlate with the activity of the neurons. And then what happens if the fly is hanging like this, it'll naturally go into a flying movement. It doesn't sense the ground beneath its feet, so it starts beating its wings. Those wing beats are captured with a camera. And let me just go back and show you this movie again. Now you can see the camera watching the wing beats. Those purple lines are the front extreme extension of the wing beat. And you could see them jittering around. What happens if a fly wants to turn is it extends its wing beat on one side or the other. And by monitoring those wing beats amplitudes, you can infer how the fly is turning through the world. And then this dot up here is like the stripe in my last lectures. It's the reference, and that is it coupled in closed loop to these motions of the wing. And so that should appear to the fly as if it's a fixed object, like the sun in my schematic pictures. So that's the reference. But if you'll notice on this virtual screen, there's also a field of kind of random dots. And those are used to induce an optic flow that makes the fly experience motion through the world. So you see that down here. You can start with this center one. If you have those dots moving outward like that, then it's like an optic flow, as if you're flying through a forest or something like that, and you see all these objects rushing by you. And so that would simulate forward motion. But you can reverse that flow like here on the left, and the fly will experience as if it's actually being blown backwards through the world, despite the fact that it's flapping its wings. And similarly, you see through these different pictures, the experimentalists can simulate drift to the left, drift to the right, any direction you want. And so in that way, the Chung in this case could look at a fly experiencing a drift as it flies through a virtual world. Now, last time I introduced you to 2 parts of the central complex. I'll remind you, the ellipsoid body is where the compass was built, and there was a hot spot of activity in that system that represented the heading direction of the fly. That's just straightforward from the head. And that was projected up to 2 copies of that signal up in the protocerebral bridge, where 2 more sort of circular layouts of angles were introduced. So we have 2 regions here that can be parameterized by an angle, and that angle, like last time, that's going to be a label on the neurons that's going to be extremely important to our discussion. Okay, so remember also the compass signals made here in the ellipsoid body, but in the protocerebral bridge, that compass signal, I didn't show you this last time, but it's very relevant for today's lecture, is going to be conveyed to other neurons so that they also know which way the fly is heading. And so that's going to be critical to our calculation. But now what I want to do is introduce you to the 3rd region that's going to be relevant to us, which is really the navigational computer in the fly's brain. We've seen the compass system, this sort of kind of switchboard, the protocerebral bridge. But now here's the computer, here's the CPU. It's called the fan-shaped body. You can see in the picture how it's kind of made into columns. It's sort of like an array of columns. And those columns, again, line up in angles not two copies, but one copy of the circle. And so again, this fan-shaped body, if you imagined, stretching it around into a circle, it's another circle. It's representing the circle again, and the different columns of the fan-shaped body represent different angles, just as we were talking about last time. And the different layers, you can see all these layers here, are different sort of regions of computation. There are many, many computations going on in this region of the fly brain. We'll talk about one of them. But across those layers, the different computations take place. And, we haven't really, nobody's worked them all out yet, but there's a lot going on in there. Okay, so we have another area, fan-shaped body, also laid out in angles. That's the basic idea here. Okay, so the first thing that we have to do, this is actually not in the order in which it actually happened in the experiments, but I'm going to do it in kind of the logical order. We have to find this traveling direction signal, the same way as last time we found these EPG neurons that were the compass neurons that contained the compass signal, the heading signal. We need to find the traveling signal. And I'll just jump to the answer. It is contained or represented by the activity of a set of neurons that you're seeing here in this picture called H delta B. You'll see why they're called H delta in a while in my talk, and B is just what they're called because there's other H deltas. Okay, so they're an array of neurons that go all the way across a fan-shaped body like this. As a result, individual neurons can be assigned an angle label depending on where they project. We can call this from 0 to 360 if you want, or minus 180 to 180. And collectively, they form a full map across that angle space. And they, like the EPG neurons, make a bump of activity, a hotspot of activity. Instead of being on a circle, it's sort of a virtual circle, but you really should think of this point here joining with that. So within the circuitry, it's a circle, even though it doesn't look like a circle. And by looking at that hotspot of activity, we can read out an angle from these neurons, the angle that they are reporting. So this is what it looks like. You see activity across a band where those EPGs are, but you see a hotspot at a certain point. And so what Chung did was to measure the location of that hotspot of activity as the fly was flying along, and he adjusted the flow pattern in all various different directions. And I will just make a lot of experimental work short by saying that when you do that, you find that this bump of activity in the H delta Bs tracks the traveling direction of the fly very, very beautifully. That's what these red dots are supposed to imply. And, this is not a subtle effect. For example, when the fly is flying backwards, this signal is full 180 degrees separated from the heading signal. So it's not like it's a subtle difference. This is definitely not the heading signal. Instead, it's the traveling signal. Do we have a question?
Yeah, we have a question. So could you repeat what are the H delta B neurons?
Yeah, okay. So they're a set of neurons that innervate this fan-shaped body. They're this set of neurons. I probably haven't shown you all of them here, but there are about 20 of them. And I'll tell you about the inputs they receive, but basically they're a set of neurons that form a bump of activity. That's what you're seeing. Whoops, I went the wrong way. That's what you're seeing here. So this is a calcium image of their activity. They form a bump of activity. And at this point, we just investigated, Chung investigated, what does that bump mean? And it turns out, as I show here, that it denotes the traveling direction for the fly. So for now, you can just think of these as a set of neurons that the fly can sort of interrogate if it wants to know which way it's traveling, as opposed to the compass neurons, which it can interrogate if it wants to know which way it's heading. So hopefully, that gives you an idea, but you're going to hear more about them as we go along, because we're going to talk about how this compass works. Okay, so here's just a little cartoon that might help, in fact, with that question. So here I've shown you from Friday's talk, this is the compass system. And that blue dot is going to face in the direction that the fly is heading, which we've called 0 degrees here in this picture. This is the H delta B. This is the traveling direction signal. And right now, when I start this little cartoon, the fly is going to be moving in the same direction it's heading. So these two guys align. But every once in a while, you'll see in this cartoon, the fly will be blown backwards by a gust of wind. And then the purple and the blue dots will separate by 180 degrees. So let me start the movie. Okay, so they're going along together, and now the fly is going to be blown backwards. Now you see this 90-degree split. Now they both turn because the fly turns, but now the fly is blown backwards. So you get the idea that sometimes they track each other if the fly is going straight, but if it's blown off course, the purple and the blue will deviate from each other. I have another movie, and I'll come back to this movie and show you the whole circuit eventually. But this is the same idea. In this movie, you see the fly at top left. The fly's heading is always going to be straight up on the slide. So the blue dot here is not going to move because the heading is fixed. But this is a little cartoon of a fly being, let's say, blown around in a swirling wind. And let me just start it. The red arrow tells you which way it's being blown. And as it's blown around in this circle, you can see that the EPG signal, that's this red signal, travels around the circle, reflecting. Now it's aligned. Right now it's going to be backwards. And the other thing depicted here is sort of a picture of the ground, because that's going to be very important. Remember, the way that the fly knows it's drifting is through optic flow. So you can see faintly there, the ground kind of moving under the fly as it drifts in these various different directions that it's going. And as I said, when I finish, I'll fill in all this cartoon with the whole circuitry of how this is computed. Okay, so the idea is I've now introduced you to a heading signal, and I talked last time about models about how that heading signal is built. And now we have a traveling direction signal, and the question is, how is it computed? So that's really what my talk is all about. Okay, so, and I mentioned that it's not magic. The fly measures its direction from optic flow. The question is how it reconstructs this vector and how it puts it into an allocentric coordinate system. So the fly measures its optic flow in this experiment. In other words, it measures this flow of random dots past its eyes in order to figure out which way it's going. And once again, in this same setup where Chung could control the flow of the optic flow that the fly was seeing, he measured a number of visual neurons. So these are now not columnar neurons. They're not in the central complex, but they're they're neurons that are part of the visual system that detect optic flow.
There's a question.
Yeah.
Is it possible to excite particular combination of neurons to make the fly fly in a particular direction?
Yeah, I'll show you that at the end. The direction that the fly was going was not the end, but you can certainly change the direction that the fly thinks it's traveling. I'll show you that right at the end of the talk.
So, and I had a question.
Yeah.
Which is that, as in the case of the flies walking on a ball, flies, they're getting proprioceptive information. Similarly, flies that are flying get information from their halteres.
Yeah.
Now the question is, in a tethered setup, halteres are probably misinforming the fly of the direction in which they're headed. So is there a problem?
Yeah, excellent question. So for those who don't know, I hope you can see my arrow. These are the halteres. They are, many insects have 4 wings. They're sort of the remnants of the other 2 wings on a fly. And they are little Coriolis force detectors that are the fly's vestibular system. And in flight, they detect acceleration in turns and things like that. And you're exactly right that they do not operate properly in tethered flight. And as a result, the compass system does not work nearly as well in flight, in tethered flight, as it does in walking, just for exactly the reasons that you surmised, which is it's missing a vestibular input. And as a result, in these experiments, if we wanted to know which way the fly thought it was heading, we actually used the compass signal, not the dot on the screen. That's because it's more reliable for exactly the reason you said, that the halteres are not properly being manipulated since the fly is just hanging there.
I see. So
yeah.
Interpreting, then, I mean, a lot of these things, we are looking at this purely as visual behaviors, and we are not.
Yeah, in this thing, it's a visually guided behavior. Unfortunately, the optic flow is all visual. And as I said, we got around the haltere problem by using the compass signal directly.
Right.
So we're looking at which way the fly thinks it's moving,
right,
even though it might have some confusion in the sensory system.
Okay. Thank you.
Okay. All right. So anyway, Chung measured these visual neurons, part of the visual system, as I said. They're just a handful of them. They measure the optic flow projected onto these axes that I've shown 45 degrees relative to the fly. So if you look, for example, here, I'll just denote. This one on the left here is measuring the projection of the optic flow or the fly's perceived velocity onto this left forward vector. And this one here is measuring it projected onto the right forward vector. So I call those the velocity left forward and right forward. So they're just doing projections. They're also a set that project in the backward direction that kind of just give another copy. I will come back to these at the end of my talk, but I'm going to ignore them for the time being because they're duplicate information. In other words, the velocity is projected twice, once in the forward direction and once in the backward direction. But we can discuss the thing for a while, just ignoring those backwards ones, and then I'll bring them in at the end. Okay, so we've got this information, we have the projections, so what's the problem? Well, the problem then is given 2 projections of a vector, can you reconstruct the vector? And that's just a classic problem. I assume many of you solved this in your first year physics class. I used to teach first year physics, and I certainly taught a lot of students how to do this. I think all of us would solve it in this manner. In other words, we give our physics student these 2 components, the forward left and forward right velocity projections. And the physics student will say, well, I'll build 2 vectors, one at 45 degrees and one at minus 45 degrees. Give them the lengths that you've just given to me, the velocities, and just add up those vectors and I'll get the traveling direction. This is about as simple a physics problem as you can get. Okay, that's pretty good. And of course, from that, using arctangents, our physics student can work out the angle of this vector, which is really what we're talking about. There's just a slight complication that we're going to measure the angle relative to this heading direction. So you have to shift it by 45 degrees, no big deal. So our physics student gets that far and feels very proud of himself or herself. But that's not quite the answer, because remember, we wanted this direction relative to a fixed location in the environment. And that means we have to add on the heading direction. So the actual answer is the sum of the egocentric traveling direction plus the heading direction. And so there you go, give the physics student 100% on this problem. And I have both taught and graded many homeworks like this. And when I did this project, I sometimes kind of pretended, well, the fly was like another physics student who handed in a homework on this problem that was extremely weird and extremely creative, because although the fly gets exactly this answer, it does not do the computation quite this way. And so the rest of my talk is to tell you how the fly does the computation. It's a way that you'll all recognize, I think, but you've probably never added vectors this way. And then show you, how the circuitry actually pulls it off. Okay, so that's the basic calculation. I'm sure, as I said, you can all do it very easily. Okay, so what does the fly do? So first of all, the fly has these same projections because these visual neurons are giving these feed forward right, feed forward left numbers to the fly. But it actually does something a little unusual, not, you could do it this way if you wanted, which is to do the rotation first. So rather than at the end, the way I did it in the last slide, the fly actually immediately rotates these basis vectors by the heading angle, okay? So that the fly immediately goes to these allocentric world coordinates and then does the addition. So in other words, the fly does the calculation a little bit probably reversed from the way we would do. That's not a big deal. The big deal is how does the fly then add the vectors? And the fly adds the vectors in an amusing way. First of all, fly introduces an auxiliary variable, which is this angle, which is the angle across these structures. That's available anatomically to the fly. But if we were thinking of the fly's homework, the fly would say, I'm going to introduce a new angle, theta. Why are you doing that? I don't know. Okay, and then introduces a function of theta, and it's this function. And I'm going to show you that in the fly's brain, this function is exactly realized across these structures. And then it takes the maximum of this function, okay? Just computes the maximum. And if you do your little homework and compute the maximum of function, you'll find it's exactly the right answer. So this is an alternate way to add vectors as an optimization problem, if you want. And it's how the fly does it. So as to say, first rotates to allocentric coordinates, then creates these functions, and then does a maximization on these functions. Okay, so what on earth is going on here? You probably have not added vectors this way. I don't think I ever had before I did this project. So let me tell you what's going on. If you take a vector in 2 dimensions, of course, it's specified by a length and an angle. And you can make a map of that length and that angle onto a sinusoidal curve by having the maximum of the sinusoid be at the angle, or if you want the phase of the sinusoid represent the angle, and the amplitude of the sinusoid represent the length. Okay, this is obviously just a one-to-one map. Now, the point of doing this is that if you have another vector with a length L2 and an angle phi 2, of course, you can do exactly the same thing. You can map that second vector onto another sinusoid where its maximum or its phase is phi 2 and its magnitude is the length 2. Now, the payoff comes that if you want to add these vectors to get a third vector with length L3 and phi 3, all you have to do on the right side is to add those 2 sinusoids. If you add 2 sinusoids, you get another sinusoid, a third one. And again, I can leave it as a little exercise for you to do a homework exercise. You will find that that third sinusoid will have its phase exactly at the angle of the summed vector and its magnitude at the length of the summed vector. So this is a way of doing vector addition. It's called the phasor method. Let me just stress to you that the idea is that there are these arrays of neurons, for example, the H delta Bs that I've shown, but I'm going to introduce some other ones, arrays of neurons across these structures that are labeled by an angle, and their activity can just map out these sinusoids And therefore, the fly can very easily represent these sinusoids, and it can add them up just by having inputs to a neuron and having that neuron sum the inputs. So I'm going to describe to you exactly how these different sinusoids are formed and where they're formed and what they look like, et cetera. But if we go back to the physics a little bit, some of you may have learned this transformation, but you would have learned it backwards. I learned it in school. That is that sometimes when you study oscillations in physics, the books or whatever, the professor will tell you that if you want to work out how to add oscillations, you map them over to vectors. So in physics classes, we go from the right of the screen to the left of the screen. Honestly, I could never figure out what this was for because you just would do it with complex numbers, but I do remember being taught it. Now, what we're saying is the fly is going backwards. It's going from the left to the right, something that we never do in our physics classes, and representing vectors as sinusoids and then adding the sinusoids. This idea has been around in the literature, both for the hippocampus, that's these first two papers, and for fly navigation in various papers that are very interesting. And I would say, the experimental work that I'm going to describe really shows that it's actually true. I remember reading, for example, this Turetsky, Reddish, and Wan paper and thinking, wow, that's really beautiful, but it's never going to be true in nature. But in fact, this system, it's exactly what it does. So that's the transformation that we're going to be talking about. Okay, so again, the angle, I mentioned the fly is introducing a function of an extra variable, and that extra variable is exactly the location of the different neurons in these structures, across these structures. And it creates the function with a family of neurons across the structure with sinusoidally varying level of activity. I'll show you that in a second. All right, so what have I got to convince you to show you this is actually true and not just a figment of mathematical imagination? So first of all, we need the optic flow to deliver us these two velocity projections, forward, left, forward, right. And then we better show that this function is actually created somewhere and that it's then maximized. I'll do that in a while. What I'm going to do step by step is to show you how number 4 is made, because it's made in pieces. It's made first as two separate functions. These functions I've shown, which are functions of the anatomical angle minus the heading angle. That's what H is here. The heading angle with amplitudes proportional to these projections. And then it's shifted. That's step number 3. And then it's added. And then finally, the maximum is taken. Okay. So I will now take you through the biology of all those steps. But I think hopefully the mathematics is all set. You build these functions, you shift them, you add them, you find the maximum, and you're going to get the right angle. So I've already showed you how the optic flow works, right? And I've argued that maximum at least is represented by these H delta B neurons, because remember, they had a hotspot of activity, that's the peak of this sine wave, at the correct angle, the traveling angle, the angle of this vector relative to the world. And so the plan is now then, I've got to take you through all these steps. So are there any questions at this point, just to make sure we're all on the same, we've got the physics problem straight before we jump into the biology? Okay, no?
No questions yet.
Okay, great. Okay, good. Okay, so we got to start building these functions. So first of all, let me just go back.
Sorry, there is a question.
Yeah.
Just came up.
Yeah, good.
Could the function of the central complex be more general? For example, vector additions could be used to maximize rewards.
Yeah, absolutely. Yes, I will get to that at the end. I'm going to do the case that, we've worked out in great detail, which is computing this traveling direction. But you're absolutely right. A traveling direction all by itself is not going to do you any good. What you want to do is compare it to a goal direction, let's say, and then you get into sort of maximizing the reward. So if I know I want to go to a certain place and I know which direction I'm traveling, I know how to turn. So absolutely, I will get to that at the end. And if you want to ask again or discuss it towards the end of the talk, that would be great. Okay, so first of all, so I'm arguing that the maximum is going to be taken by these H delta B's. And in fact, the summation is also done at the H delta B, because these functions here in number 3 are going to converge as inputs to the H delta B's and be added together. So really 4 and 5 is handled by the H delta B. But who makes this function? Now, if we think about whatever neurons make this function, they have to have access to 2 pieces of information. Let's start at the top. They have to know the heading direction of the fly. So they've got to tap into the compass system that I described on Friday, read that out and transfer it into this function. And that's how they go from egocentric to allocentric coordinates, okay? So they better hook up to the compass. The other thing is they better know these numbers, and these numbers are given by these visual neurons, right? So they better hook up to those visual neurons, otherwise how are they ever going to get that information? And so what you're seeing here is this transformation of the visual information, which is measured in egocentric coordinates because it's measured by the eyes that are attached to the fly, being transferred into a world coordinate system. So anyway, these neurons had better do 3 things. They better couple to the compass neurons, they better couple to the visual motion neurons, and they better couple to these H delta B's, because ultimately they've got to deliver that signal to the H delta B's. And there is a class of neurons that satisfy exactly those criteria. And those are the guys who are going to represent these functions and therefore represent the vectors. They look like this. This thing is the protocerebral bridge. Remember, that's the switchboard where the compass signal gets transferred to a lot of other neurons. And you notice these neurons have a nice big projection up there. That's where they get the heading signal. They connect with the compass neurons and pick up the heading signal from the compass neurons. Okay, so they've satisfied criterion number 1. Okay, what about this? This is the nodulus that I mentioned very briefly at the beginning of my first talk. It's not going to come up again, but you notice that within that nodulus, it's connecting to this green neuron, and the green neuron is exactly one of those neurons that reports in its activity the projection of the motion, optic motion. So this is where the visual motion information comes in. And so it's great. We've got the heading direction and the visual motion converging on a neuron. And finally, if you look at that little stub there within the fan-shaped body, that is where they connect to the H delta B's. So they have exactly the capacity to combine these 2 signals and deliver it to the H delta B's, which is what we need to do this calculation. And we know all this from the connectome. We can even count the synapses in all of these places and make sure that they're actually strong connections, for example, between those things. I'll come back to a little detail of this connection in a little while, but that's the idea. And like all of the neurons I've been discussing, there's not just one of these, they tile these structures. So they have another property, which I don't mention, which is they have to be able to represent a function of these angles defined across these structures. And they can only do that if, as a population, they tile. And so, you know, there's holes here just because I didn't make I got tired making the picture, but they completely tile these things. And there are 2 sets shown here. There's a left set and a right set, and there are actually 2 more sets that I'm not showing. So there are a total of 4 sets of these, and those you'll see in the end, those 4 sets correspond to these 4 black axes, the forward left and right and the backward left and right. So they are built to represent those vectors. And they, as I say, they have the right properties to do that. Okay, so how do you make this function? In addition to just having those properties, this has to be a cosine, okay? This isn't, you know, last time on Friday, I kind of used a cosine as a generic function just because it was easy to calculate with. This is not arbitrary anymore. This really needs to be a cosine because this map to the phasor representation only works for cosines. It's a property of the cosine angle addition formula that makes this work. So the profile of this set of PFN neurons across these structures had better look like a cosine. That's an experimental prediction, if you want, that can be tested. It better move with the heading angle, right? So this is a function that peaks at the heading angle. It better move back and forth. Its peak better move when the fly turns. And so, you know, how do we get those things right? I'm going to start with those features. So I mentioned before, you know, already that you have these copies of the heading direction angle that are built in the donut-shaped ellipsoid body, but projected up to the protocerebral bridge. And I just want to remind you what that signal looks like in the protocerebral bridge. We can now forget about the ellipsoid body because we're now moving up to the protocerebral bridge and then down to the fan-shaped body. So just assume the compass has been built. That was the last lecture. And just let me remind you that we would see shapes of activity if we looked at the compass neurons now that would move back and forth across the protocerebral bridge as the fly turns. That's the angle, that's the analog of the compass turning around in the circle of the ellipsoid body. But these shapes are not sinusoidal. They kind of look like what I've drawn. And they don't have to be because this is not directly involved in vector arithmetic like these other neurons. Okay, so what Chung had to do was to look up in the protocerebral bridge at the activity of these PFN neurons and plot it out across the bridge. And the answer is that it's beautifully sinusoidal. Unlike the EPG signal, it really fits very nicely into a sinusoid, meaning that these different compartments, if you look, remember the different compartments contain different PFN neurons. And if you measure the activity in those compartments, like these dots, and plot them up, you find, you know, a discrete approximation, if you want, to a sinusoidal signal. So it really is a cosine, and it really is quite accurately a cosine. Now, how did that happen? I mentioned last time this delta 7 neuron, and our belief is that this delta 7 neuron is specifically built to make this thing sinusoidal. I talked last time about how it has a sinusoidal profile of its connections. And so, you know, this is, I just think it's beautiful. It's nature realizing that cosines are special or sines are special functions, that they have special properties that are useful to getting around in the world, and building that into the anatomy of a neuron with this sinusoidal density of synapses that I showed you last time, sorry, in this structure. So this is nature, if you want, building a table of sinusoidal values in order to look it up when it has to do calculations, just the way you know, you would, although nobody uses tables anymore. Okay, so we've got our cosine. Now, what about, does it track the heading direction? So again, Chung just had to measure the PFM just, you know, that's easy for me to say, but had to measure the activity of these PFN neurons. They're the vector representing neurons, and the compass neurons, the EPG neurons, as the fly turned and the EPGs moved, and sure enough, they all moved together. So they're all locked to the compass signal. So what does that mean? what that means is we built our cosine functions, right? Not only are they cosines across the angle, remember theta is this angle or this angle. We've got two sets of cosines, the left one and the right one. So not only is it a cosine, but it shifts. I'm not showing that here, but it shifts with the heading of the fly. So if the fly turns one way, this cosine would shift this way, shift that way. And so it's exactly a cosine function, just what we wanted, except it's not shifted by 45 degrees, but we'll get there. And so we've accomplished our first step. But remember, in the representation of a vector, there are two things that have to be required. First of all, the phase here should be the direction of the vector, but the amplitude should be the length of the vector. And so we need to make sure that the lengths are the appropriate lengths, namely the projections of this vector onto the left and the right 45-degree angles that the visual system does. So Chung had to go back and not only measure that it's a cosine profile, but that it's modulated, the amplitude of that cosine. So that would be this brown cosine getting bigger and smaller, depending on which way the fly's optic flow was drifting in the experiment. That's the idea, and you'll see that in a second. Okay, so we're looking for those terms in the computation. Okay, so that's the idea. Put the fly back into the optic flow and measure these cosine profiles. And here they are. So this is now directly data. So this is the cosine profile, sinusoidal profile, whatever you want to call it, of the right-hand PFNs, and here the left-hand PFNs. So this is directly data. These are going to be centered, so they're always centered, so corrected for the heading of the fly. So it's as if the fly was always moving in the heading direction. And you know, here you see when the fly is moving forward, you get, you know, two nice big sinusoids. Now, if this were true, then you ought to get exactly this result. So when you do exactly the same measurement, but the optic flow is tricking the fly into thinking it's moving backwards, then suddenly there's still sinusoids there. The activity profile of these neurons is still sinusoidal, but the amplitude is greatly reduced. That's the idea, that they are reflecting the projection of this vector. It's actually offset. They're actually measuring a constant plus the projection of these vectors. That's why this doesn't go negative. It just
There's a question.
Yeah.
The output, how do the delta 7 neurons affect the output sinusoidal activity? Is it because of the structural compartmentalization of the protocerebral bridge, or are the neurons doing some sort of a computation as a result of their interactions with these compartments?
Yeah, very good. I didn't explain this very well now, I realize. So most of the EPG signal to these PFNs, in other words, the compass signal goes through the delta 7s. And what happens is the delta 7 is like a Fourier transformer, basically. It's whatever it takes in, it spits out a cosine version. So it's as if it was computing the first, you know, Fourier component of the signal you give it. So, it's like a Fourier decomposition. So it takes the compass bump, it doesn't really matter what shape it is, and it spits out the best matching cosine to that compass bump. Hopefully that answers. So that's the computation that's being done. Yeah, I realize I should explain that more in the talk. Okay, any other questions now?
So I had a question. So, you know, these are being measured as calcium signals from these neurons, but are they spiking neurons, or what kind of neurons are they?
Yeah, so they are spiking neurons. We don't have, you know, intracellular recordings of these, so their spiking activity is not well known, certainly not in these experiments. That was not done. So what you'll see is this signal definitely gets transmitted to the next stage. So whether it gets transmitted in a graded manner or spiking manner, I don't know, but it is faithfully transmitted.
Mm. Okay. Okay, thanks.
Okay, yeah, good question. Is there another question? Maybe another? Or we all set?
No more questions.
Okay, all right. And so likewise, if you measure, for example, here the fly thinks it's drifting to the left, and sure enough, the left sinusoid, which should be proportional to that drift velocity, gets big, and the right one, which now has a very small drift velocity, gets small. And you can do that across the whole range, measure these amplitudes, and you find they're exactly equal to the forward left and forward right projections of the optic flow. So this is a kind of a beautiful transformation. I showed you curves that look pretty much like this before, but those were the activity of visual neurons, of individual visual neurons. That signal has been transformed to the amplitude of a pattern of activity in a whole population of neurons. So this curve now on this plot is not the activity of an individual neuron, it's the amplitude of the shape of the activity of a population of neurons. So a very tricky transformation has been done. And the point of that transformation is to change from egocentric to allocentric coordinates. Because I think I have that on the same fly, you've taken this information and now multiplied it by a vector that moves with the, by an angle factor that moves with the heading direction. So these two, according to the phasor representation, these two terms represent two vectors, one that has the length of the forward right projection and one that has the length of the forward left projection, and both of which point in the heading direction. So we have a problem here. I'll do it with my hand since I didn't make a diagram, in that the vectors we want to represent were off at 45 degrees, right? They went like that. And they had lengths that were, V forward left and V forward right. We've almost got that. We've got the lengths right, but now these two vectors both point in the forward direction. They're both pointed in the heading direction. So we've got two vectors represented, but they're pointing the wrong way. And so that needs to be fixed. Okay, and that's fixed in the next line. Okay, so in other words, we have these cosines that are both h, and we want to move them by plus and minus 45 to get them back pointing in the right directions. And that's what these pi over 4s are. So we take these two things and rotate them in order to have them pointing in the direction, that they're supposed to point. And so this is where you see that if this h wasn't here, this would be a phasor representation of the egocentric coordinate term. It would be at plus and minus 45. But because it's picked up the heading direction, it's now these two vectors in a world reference system, reference relative to the heading. So this does the coordinate transformation, and then this takes those two aligned vectors and moves them into the right angles. So we've now represented the vectors properly. Okay, how does that happen? Well, I mentioned that so far we've been talking about activity up here in the bridge, which is where the heading signal gets picked up. The velocity signal that you saw reflected in the bridge gets picked up way down here in the nodulus. I haven't shown it here. And in answer to that spiking question, it gets conveyed up, beautifully up to this level. So there's certainly a transmission of these signals. Again, I don't know if it's spike mediated or not. Probably spike mediated. Okay, so we've been looking up here, but I mentioned that the actual calculation is done down here, and these H delta B neurons that are going to display the answer of all this calculation, they live down here. So we've got to project that signal down. And fortunately, as I showed you a little while ago, these PFN neurons do in fact project there. In fact, PFN stands for protocerebral bridge, fan-shaped body, and nodulus. So they project to all three, and so they project down. And when they project down, a very cool thing happens. Remember, we have to shift these angles by 45 degrees. We have to take two vectors that are aligned and do this. So if you look at this connection that I told you about before, the connection to the H delta B, it's actually in the wrong place. So I mentioned that, you can't see it on this diagram, but let me show you on the previous one. Right, there's a one-to-one correspondence between regions here and regions here. They correspond in angle, and we can match them up. But if you look carefully at this diagram, I mean, you can't see it, but you have to take my word for it. What you find is that this output is misaligned. If this had gone from the correct compartment here to the correct compartment here, it would have ended up here in the middle. And instead, it's shifted over. And that shift, remember, this whole thing is 360 degrees, so you can kind of estimate it. That shift is exactly 45 degrees. So basically what happens here is these PFNs that create this function up in the bridge, when they project down, they shift. And they shift over by 45 degrees. So that when they make their sinusoid down in the fan-shaped body, it's shifted and it becomes exactly these functions. So, again, I think, the beauty of this and the way evolution has transferred mathematics into anatomy is really, very beautiful. That the system, as I mentioned, it creates a table of a cosine in these delta 7 neurons. It creates a multiplicative interaction in order to be able to represent vectors in this phasor way. And then anatomically, it creates this 45-degree shift that aligns up these vectors properly with the shift that's in the anatomy of the visual system. I also mentioned, last time we had another 45-degree shifts. If you remember right, these PEN1 neurons, the neurons that allowed the bump to turn when the animal turned in the absence of visual input, they were also a shift by 45 degrees. And I think somebody asked, why 45 degrees? Yeah. And I mentioned in that case, it didn't really have to be 45 degrees. It just had to be off to the left and off to the right. In this case, it really needs to be 45 degrees, or it basically has to be whatever angle the visual system chose. The visual system happened to choose 45. So this case, the 45 is really for real. It's what it has to be. And we even checked in the EM anatomy to make sure that this angle really is 45 degrees. And my assumption is that the PN1, just let me finish this and I'll answer. The PN1 uses 45 degrees just because the anatomy had figured out how to make 45 degrees. You have a comment or question?
Sorry. So then the hypothesis would be that if there are flies in which have eyes differently situated, which are not, which don't have the 45 degrees, then you would see it, this angle is changing. Is that?
Yeah. So yes, exactly. You would, for example, another species or something. That if another species didn't happen to choose 45 for its projection, you're exactly right. This angle here would change. That would be the prediction. Huh. So you'd have a very, very strong anatomical prediction.
Cool.
Okay. All right. Are we there? Then let me go on. Okay. So we have now accomplished this step. We've created the two functions that represent the two vectors. And we're basically there now. We just need to add them up and find the maximum. And they are added up because they are both inputs to H delta B. So that's trivial. They just all converge onto H delta B and get summed as separate inputs to the H delta B. And the H delta B, we believe, has a self-sustaining bump. So I'm now getting to the maximum. Sorry, so we've summed these angles up, these functions up. How do we now find the maximum? So the idea here is just like the compass system, the H delta B has a self-sustaining bump that could exist anywhere. So I didn't use this terminology so much on Friday, but the idea is these systems build a line attractor, a ring attractor actually. A ring attractor means that you can have a bump of activity anywhere on the circle with equal likelihood. There's a symmetry in the circuitry so that, for example, this purple dot that I've drawn here could equally well be happy to sit at any place along that arc, which is really a circle. But if there's an input, so it can be, anywhere. But if there's an input like what I've shown, in other words, when you add up these two sinusoids, remember you get another sinusoid whose phase is at the resultant of adding the two angles, of adding to two vectors, I'm sorry. So this is the traveling direction. And if you have a bump like this, which is created by this function, then the symmetry is broken. This is just like the visual input term that I discussed last time. In this case, it's an indirect visual input. It's a highly processed visual input that signifies the sum of these two traveling, components. And so the bump is going to sit at the peak of that function. So if you want, ring attractors are a great way to do optimization because they'll automatically move to the maximum of their input. So if you want to maximize a function, you just put it on the input to a ring attractor and look at where it goes, and that's the answer. That's the maximum. So that's how the maximum is computed. And we got it, right? That maximum is going to be at the allocentric traveling direction because, I told you that when you maximize this function, you get the right answer, but also because we're basically doing vector addition, just like our physics student, just in a kind of a funny way. I would give this student 100% for getting the right answer and a plus for creativity, I think, if I'd seen it in a physics course, and maybe told him to go see a counselor or her to go see a counselor about being a very weird person. Okay, so we've got it. We've calculated everything, and there it is. There's our signal. We've accounted for exactly how it's computed, except that I cheated a little bit. And so let me just finish by, and then we should have plenty of time for discussion actually, because I can finish up in not too long a time. So let me just remind you though, that I've been discussing these forward projecting reports of the optic flow. And I didn't mention, but these come from neurons called LNO2 and SPSP. Those are the visual neurons. There are a couple of them on each side. So small number of neurons, not columnar neurons, just individual neurons that fire at a rate that reports these two forward left, forward right projections. But I mentioned quickly that there's another set, LNO1, another set of neurons that reports the backward projections. So it's as if the fly took this coordinate system and instead of dividing it into two axes, divided it into four axes and reports four numbers: the forward left, forward right, backward left, and backward right. I'll give you a tidbit from mathematics history. We all know Descartes introduced the Cartesian coordinate system. When Descartes did it, he did it like the fly. Descartes actually proposed using four axes and four numbers to denote a position or a vector in two dimensions. And it was actually Newton who said, we don't really need to do this. We can use negative numbers to represent, and we really only need two axes. So this has an interesting history in mathematics as well. Okay, so really what we're doing here is we're adding four vectors, but they're kind of duplicates of each other. So first of all, why is the fly doing this? There's one reason that makes sense, and I kind of skipped over it and nobody caught me on the question period. But if you remember, the neurons that were encoding in their amplitude these components, they did it with an offset. In other words, those amplitudes were always positive. So rather than reporting, let's say, V forward left, it was really a constant plus V forward left. And that constant, you would think, would provide a forward offset, right? It would be as if there was always a constant projection onto both of these axes. So you would think that would give the impression it's constantly moving forward, the fly. Now, when you have the backward vectors, they're also offset by the same constant. And when you add up the 4 vectors, the constant cancels because you have a constant representing flow forward and another constant backwards, and they cancel. So this allows the system to work with an offset, so it doesn't have to work with negative amplitudes or negative firing rates, for example, here, and very neatly cancels this offset, which is why I didn't talk about it, because in the end, it works itself out. So in this case, there was a real reason, if you want, like Descartes, the fly really doesn't want to use negative numbers because in its activities, it cannot represent negative numbers. And so it does this double representation of the forward side and the backward side of its motion. Okay, so how does this work? Same old way, it's just we have to represent 4 vectors instead of 2 vectors. And those 4 vectors are represented by these 4 sets of PFN neurons. If you want, my talk thus far was only about these 2 in darker brown, the PFNDs on the left and the right. And I just ignored the backwards ones. But there's another backwards set that do exactly what I was telling. There's only one little wrinkle that I have to tell you about. They compute it in exactly the same way. I think I actually showed that. And these vectors, let me just show you this diagram, show really nicely. You might say, well, you're representing the same thing twice, but you're not, because if the fly turns, these vectors are attached to the fly. They're part of the visual system of the fly. But these other vectors stay fixed in space, right? Because they rotate with the compass. And so you've gone from a system of coordinates that moves with the fly to a fixed set of coordinates. And if Chung does the imaging, I'm just showing you quickly that it works just like I told you. What you find is both the forward set and the backward set move together. They're all sinusoidal. They vary with amplitude in exactly the right way so that the backwards one represents, if you notice, they're exactly backwards from the forwards one. So they represent the backwards vectors. So it's the same story I just told you all over again. The only trick here is that the backwards vectors have to point at 135 degrees, right? I showed you how this 45-degree plus and minus gets represented through the shifts. Now, this bothered us a lot because these PFN Vs, they also shift by 45 degrees, but they represent vectors that are rotated from the heading by 135 degrees. So where does the extra angle come in? It was Gabby who figured this out. How do you get to 135 from 45? And that is, if you look at the anatomy, I didn't show you this before, I showed you all the H delta B's together. If you look at them individually, they have this funny structure with 2 connection points, one here and one here. And what you find is that the forward vectors connect up there, but the backward vector representing neurons connect there. And the difference here is exactly 180 degrees. You can see that. It's exactly halfway around the arc. And so what happens is, in addition to the 45-degree shift, there's an extra anatomical 180 shift. You add it up, you get your 135. So again, the anatomy here is just, to me, it's just spectacular that this system has built into it angles like 45, 180. There are also 90-degree shifts that are not part of this talk, but there's 90-degree shifts too, which is a way you can turn, let's say, a cosine into a sine. So the anatomy here, the fly through evolution really worked out trigonometry. It figured out all about angles and cosines and sines. And so the ultimate calculation looks like this. Our physics student would take 4 vectors with X and Y components given like this, and amplitudes given like this, and add them up. And the fly is taking 4 functions, shifted appropriately and modulated appropriately, and adding those up. Okay, you can build a model of this system. It's kind of the easiest model you can imagine. You just take the PFN activities and add them up, basically. And we did that with 2 assumptions. One is to add them up with assuming that every vector counts as the same, the way I just showed you. That's an assumption. We don't know it for sure in the fly anatomy. But when you do that, you get these red circles. So this is a plot of the H delta B. This is just showing that the H delta B follows the traveling direction. You know, the error bar things are the data. The red circles are this model where we guessed equal contributions, but the green diamonds are actually using the counts of synapses. So we didn't even have to assume equal contribution. We could actually use the synapse counts to weight the different things. And you can see that model works very well as well. So basically, this is a 0 parameter model that matches the data. Okay. I'd like to leave a little time for discussion, so I'm going to do this fairly quickly. But you know, I just want to get the flavor of this, because in fly research, when you have great experimental collaborators, as I did, you can do the most amazing manipulations once you have an idea like this to try to verify that it's really true in a causal way. And so by expressing various different proteins in the cells, in the different cells. Shibiri, for example, blocks synaptic transmission. You can manipulate this circuit and see if it does what you think it ought to do. So the first manipulation I'm showing here is to block the compass. So when the compass gets blocked, the system should kind of work, but it gets unmoored from the real world. So the traveling direction is just going to sort of flap around in the world and be unmoored. And again, I'm not going to go through in detail, but that's exactly what happens. The compass signal is still there, but it's just untethered to the world when you do that with one of these genetic manipulations. You can also manipulate the vectors. So you really can do a little vector arithmetic with optogenetics. So in this case, what you do is to inhibit the backward going PFNs. Remember, there are 2 PFNs that represent the forward direction and 2 PFNs, the PFNVs that represent the backward direction. If you inhibit them, it's like you shorten the backwards direction. And it's as if now this balance that I talked about is screwed up. And now the fly should feel like it's constantly going forward. And indeed, that's what happens. I don't know what the fly feels like, but the traveling direction signal starts pointing in the forward direction. You can excite the backward direction, and then you have a fly that feels like I'm sure you and I do on some days, like you're just going backwards all day long. And so again, I don't know how the fly feels, but the compass starts pointing in the backwards direction. And you can also inhibit the front going vectors, and again, get the traveling direction signal to point in the backwards direction. Okay, so let me give you a summary of the whole thing and then a little future directions, and then I'm happy to have a little discussion, take other questions or whatever. So again, we started out with a fly drifting around in a breeze. And this is going to be a fly, for simplicity, the fly is always going to be headed in the same direction, but it's going to be blown about by, if you want, a circular current of air, so that sometimes it's going to the right, sometimes it's going backwards, sometimes it's going to the left, like this. All right, so what have I argued? First of all, I've argued from the fly's point of view, the optic flow that you're seeing underneath the fly, as if it was a pattern on the ground, is projected onto these 45-degree axes, not the up, down, and sideways axes on the left. is projected like that. And that motion affects the activity of visually responsive neurons, the LNs that go into the nodulus, and this SPSP, which actually goes into the bridge. So you can see them being modulated up and down by the drift and the optic flow. Now, as I showed you, that gets translated into the amplitude of sinusoidal patterns of activity in the procerebral bridge of these PFN neurons. So they go up and down in this pattern that I'm showing you as the fly is drifting one way or the other. Those get projected down to the fan-shaped body as 4 sinusoids across the structure of the fan-shaped body. That's what you're seeing here. And if you add up those 4 sinusoids, you get the red sinusoid, which is the vector sum, and is a sinusoid that represents the vector of traveling direction. And finally, the bump in the H delta B just rides on the top of that red sinusoid and reflects the maximum, and therefore goes around in this circle to represent the traveling direction of the fly. Now, I mentioned already, so I'll just be quick about this. Clearly, the next step is to find how goals are represented in this system, because we would think that you would want to compare your traveling direction to some sort of internal goal and perform turns appropriately to make you move in the desired direction. So that's underway, and to generate a turning signal. In this paper, Stone et al., that I already mentioned once, they use the same kind of phasor representation to show how you could do path integration. Basically, if you can add vectors, you can integrate vectors. And if you can integrate vectors, you can integrate your motion along a complex path like this. And for example, in this paper, they use these same methods to calculate a vector that always points back to your starting point. So if you're a nesting animal like an ant or a bee that goes back to a home, you can keep track of the direction home along a convoluted path as you fly around. Using calculations like this, now exactly how this happens within the anatomy of the fly, we don't know yet, but you could stay tuned, because I would predict that both of these things are going to be worked out over the coming years. We now have all the tools, we collectively, that we need to do this. Finally, I just remind you that this is all part of a system that I think doesn't just do navigation, but really tells the fly how it's oriented relative to the world, probably is involved in all of the motor actions that the fly takes. You know, if I start waving my hands around to explain something to you, I know where my computer screen is. I know not to reach too far and to slap the screen. And so every motion I make is really relative to the world around me. And I think even that's true for flies. You know, you don't want to extend out your wing and smash it into something. And so, so this is part of a general system for kind of keeping track of the world. Okay, so good. We have a little time discussion. I'm happy to take comments, questions, discuss really anything about the broader implications, whatever you want. Maybe I'll stop sharing and I can look at your faces.
A couple of questions. Wait a minute. Have these methods been used to analyze the dance patterns of other insects? Like why those insects use those specific patterns and not any other pattern?
Yeah, so I'll tell you, that would be my dream. If I could, in this research, you know, call up anything that I wanted, it would be to look, suppose we find a goal representation, for example, which I think is possible. So in a bee, you would have bees standing around in the hive. A bee would come back and do this waggle dance, and that goal direction would get impressed into their heads. So this would be an opportunity to actually see communication, you know, going into a brain and being represented in a brain. It would be mind-blowing. The trouble with that is that bees are just difficult in many ways to work with. The queen structure makes the genetics much more difficult. I don't know whether that's going to happen, but you know, it's cool to think about that we're kind of, we could imagine doing such a thing, which I think would just be amazing. In the same way, you know, if I told you, if you want to get to my house, you got to go 3 streets and turn right. Yeah. Clearly a map is being generated in your head. Yeah. In bees, we could maybe really watch that process happening. So it's a cool thought. Unfortunately, it's just a dream at the moment.
Okay, so Vatsala has a question, then Vijay, and then I'll ask a couple more questions. Vatsala?
Yeah, hi. So I have 2 half-formed questions. Maybe it counts as one question. So one question is, what is the role of the motor efference copy in all of this? Presumably, fly not only has access to optic flow, but also, you know, receives what a plan of the movement to be executed, and presumably uses that in navigation. That's question number 1. And question number 2 is, you spoke about Hebbian plasticity in connecting these circuits in the ellipsoid body on Friday. I'm just wondering how hardwired are the connections in the fan-shaped body, and whether, you know, there is plasticity in connecting that up at all. And if not, then there's obviously a conflict between, you know, plastic structures versus you know, non-plastic hardwired structures, and how to resolve those 2.
Yeah, both great questions. So first of all, you know, I mostly talked about proprioception, but even yesterday, the idea that when the fly turns, the compass turns, that must be motor copy as well as proprioception. And we don't know to what percentage. The same thing here. I'm sure the effort that the fly is exerting on its wings plays a part of this. One thing that may come up from somebody's question is that I've completely ignored the speed of the fly here, if you notice. Now, Chung did some measurements at different speeds, different optic flow speeds, and we could not see a really good representation of speed. Speed was affecting things, but it was kind of a mess. And I think that may be because we couldn't monitor this fly's sense of its own speed. How hard are you beating the wings? So that's unfortunately a missing component that probably limits pieces of this. Plasticity, absolutely. So for example, for path integration, you've got to integrate the activity over the whole course of your thing. To set a goal, you have to do plasticity. And I imagine the setting of a goal would be a lot like what I talked about yesterday. You know, if you want to set the compass, you walk into environments, you sort of take a snapshot of the world and use it. I think the same thing for a goal. You know, you see a target or something, you set a direction, plasticity puts it in there. So undoubtedly plasticity is going to be a big component. So on top of the fixed anatomy that sets the angles, you'll have, you know, factors, multiplicative factors. It's a little like the velocity v left and v right thing, you know, that can go up and down. That went up because of an input, but you could imagine similarly that going up and down as a result of a synapse changing, right? Because that's part of an input. And in fact, that's how the path integration works. That amplitude factor just steadily goes up as you go further and further, and the vector, you know, starts summing up. So for sure, you know, that will be a big part of the future.
Thanks, Larry.
Yeah.
Yeah, Larry, fantastic talk. Your comment about evolution having turned mathematics to anatomy was really, really nice. I have 2 questions. So one is, what about the third direction, in the sense that what you talked about was basically the yaw, the pitch and the roll? Are there other neurons which also sense gravity and tell the fly to correct it?
Yeah, there certainly is a sense of gravity that's known in the antenna of the fly. So these things, so Chung did look at the optic flow and made it go up and down. right? Which, you can do that in this experiment, and it did not affect these neurons, none of them. So this is, what do you call this direction is, that's yaw, right?
That's yaw, yeah.
This is elevation, yeah. So this is a yaw system. There's got to be an elevation system. We don't know where it is at the moment. But, it's a little like eye movements in mammals. They're separated, right, in the two directions. So somehow this is separated for reasons, I don't know why, and I think it won't be clear until we find the up and down system.
Yeah, even in birds, they're separated. They also fly.
So it's separated.
It's cardinal directions, yeah.
So unfortunately, I don't know where that system is.
Okay. The other question that I had, maybe it's not well formulated, is that if you're trying to combine signals from two different inputs and trying to add them up, somehow the adding system should have sort of linearity property, like adding vectors. Is that obvious in the way the signals come in and they will add up linearly?
Yeah, so good point. The summation of the input has to be linear, but then what happens to that can be nonlinear here. So you're finding a maximum. So if the function gets distorted, as long as it's monotonic, it's not going to hurt it. It's true that these signals have to be summed linearly, and because they come at different places on the dendrite, on the structure, it's not obvious that would have been true. I think it has to be true. So you'd have to go into the biophysics of those cells to figure out why that's true. But once they get summed, you can then go through a nonlinearity, like a spiking nonlinearity, and that's not going to cause any problem.
Okay, thanks.
Okay, so there are some questions. Do you think multiple vectors from memory are added together while path integrating? Or is path integration done online by adding pairs of vectors while navigating? The latter option seems easier to implement but less flexible.
Yeah, so the model that I mentioned in this Stone et al. Paper, Barbara Webb is kind of the leader, the theory leader of that, and Stanley Heinze was experimental. It works in the latter way you saw. Just online, you just keep adding the vectors. There's got to be kind of more of that. You've got to reset. You've got to worry about saturation. So, I think it's probably a combination of the two things that you said. I think what you said is exactly right. Online and just adding is easier, but it's less flexible. For example, it might saturate. And so there's got to be a control system that can reset the integrator at appropriate points. For example, maybe you go to a location where the scene is distinct. You reset the compass, and at the same time you reset the integrator. I don't know, but the models that exist now are of the just online, just start adding it up kind.
Okay. So the fly uses a system of vectors, including the heading and landmarks, to compute travel direction. But how is the heading vector computed at all? Is it also a result of a maximum of directional neuronal activity of perhaps neurons that contribute to spatial orientation and motivation? Or is there something else going on?
Yeah, so I mean, that's what I really talked about on Friday. You can think of it as a maximization operation too. It's not so much a vector operation. It's more of a integration of self-motion and a pattern matching with the environment. So we wouldn't describe that in those vector terms. As I say, it's like a template matching, I think of. So you have a certain, if you talk about the visual world, you have a certain pattern impressed on that, and you can note your relation relative to that template. But I would say, that system's pretty well understood, probably better understood than this one.
How would the fly use this method of computation if it were moving against the wind and not along it? Would it remain the same, which I guess is not, or does the fly use additional mechanisms to understand that it is moving against the wind?
Yeah, so wind is a tricky business here. I guess, Kathy's talking to you about wind. So when you're in the air, you don't sense the wind. The wind you sense is your own motion through the air, but you're in the reference frame of the air. So for a fly to detect wind direction is a tricky business. And so this kind of drift is one way to do it. So, a fly can sense, let's say that it's flying into the wind by comparing how hard it's flapping its wings to its ground speed, or the kind of thing that I was showing. So by the way, this is not a fly flying into the wind. This is a fly being blown around by the wind. They can compute wind direction by looking at this optic flow and comparing it to their own effort. And in fact, people in Gabby's lab are looking at a system that could compute wind. But I think it's important to remember, especially, for Kathy Nagel's talk, that when a fly starts flying, it does not have direct access to the wind direction. It has to compute the wind direction. And so, and I think what flies do are twofold. As they take off, they remember the wind direction, and then they do some of these calculations looking at how they're drifting around to infer the wind direction.
There's this phenomenon they called optomotor anemotaxis. Is that correct?
So you're saying motion into the wind?
Yeah, I mean, that's what it's been called in literature.
Yeah, I don't know.
Anemotaxis.
You know better than I do. I looked up the bicycle paper, by the way. It's good.
Yes.
It's very good.
Good. There are no further questions. Just wait if there's any more questions. Or else we can stop here. It's just perfect. 6:30 Indian time. So thank you very much, Larry. That was an absolutely stunning talk, both of them. And this has been a lot of fun. I feel updated.
Okay. Well, thanks everybody for very good questions. It was fun to do.
Thank you, Larry. Thanks for doing this so early in the morning.
Okay.
Thanks, Larry. Wonderful talks.
Bye-bye.
Bye.
Okay, we then take a break and come back at 7.
At 7, yeah.
For Kathy's talk.
That's fine. Yeah. Okay.
See you later.
See you in a bit.